Assalamu alaikum dear friends. In this video I am going to talk about the lysosomes. Who discovered the lysosome and who coined the term lysosome? What is the polymorphic or polymorphism of lysosomes? And what are the different functions of the lysosomes? Keep watching the video up to the end. You'll get all the right kind of the information regarding the lysosome. So let's get started. Lysosome, here I have mentioned on the board earlier that the lysosomes are the cell organelles which gets pinched off from the concave surface of the Golgi apparatus. Pinched off from concave side of Golgi apparatus. I have told you in earlier classes the Golgi apparatus have two faces like this. This is the concave face and this is the convex face. Through this convex face, the vesicles comes from the endoplasmic reticulum that gets attached with this face. Then the processing of the material takes place and the processed material comes out in the form of Golgi vacuoles, and some Golgi vacuoles form the lysosomes. So lysosomes contains the enzymes. Remember these points. So the lysosomes gets pinched off from the concave side of the Golgi apparatus, and this Golgi apparatus, when we talk about it, it is found in Golgi apparatus is found in all animal cells. It is found in protozoa, protozoans. It is found in fungi, and it is found in some plants, but these lysosomes are absent in case of prokaryotes and mature mammalian RBCs. Remember this point as well. First of all, this lysosome was discovered by Christian Dedew in 1955. But the term, so far as the lysosome is concerned, it was given by Novikov. Remember this point. The number of lysosome varies from cell to cell. Cell to cell it varies, and species to species also it varies. And as far as the size of lysosome is concerned, size of the lysosome usually is found between 0.2 micrometer to 0.8 micrometer, and it is spherical in structure. Then another point, so far as the lysosomes are concerned, lysosomes contain 40 enzymes which have hydrolytic nature, means they have digestible property, means they digest the macromolecules into the simpler ones. Contain about 40 enzymes which have hydrolytic nature, and these enzymes include acid phosphatases, sulfatases, they include lipases, they include nucleases, proteases. They include etc. The enzymes which have hydrolytic nature are in the form of acid phosphatase, in the form of sulfatase, in the form of lipase, in the form of nuclease, in the form of proteases, in the form of glyoxidases. These all together are responsible to digest the various kinds of the macromolecules and the smaller ones. So remember this point as well. And when we talk about these enzymes all together, they are also called acid hydrolysis. They are called the acid hydrolysis. Why so? Because they are active in acidic medium. Means they work in acidic medium, and the acidic medium is 4 to 5. Means its pH is 4 to 5. Remember this point. Once again I will tell you, these enzymes are collectively called the acid hydrolysis because they work in acidic medium, and the acidic medium is created by the protons which are pumped inside the lysosomes. Remember this point. The protons are pumped inside the lysosome from the cytosol. So when the proton concentration goes up or gets higher inside the lysosome, then there is a creation of acidic medium. So these all kinds of the enzymes collectively called the acid hydrolysis work in this atmosphere. That is the reason they are called the acid hydrolysis. I think you got this information. Lysosome contains these kinds of the enzymes. The food material in the form of a vacuolar shape goes inside the lysosome. So the food material is digested inside the lysosome. Means if the lysosomal wall gets broken down, the hydrolytic enzymes comes out into the cytosol and it kills the cell. That is the reason I am mentioning the another point. Means if there is injured cell or dead cell, what happens? The membrane of lysosome gets ruptured. Then what happens? Hydrolytic enzymes comes out and kills the nearby cells. This is called post-mortem degeneration. I hope you got this information. What is post-mortem degeneration? Means when there is a ruptured cell in the form of the we can say the ruptured cell or the injured cell or the dead cell, the membrane of the lysosome gets ruptured and the contents comes out. Their enzymes kills the cells which are nearby this injured or the dead cell. This is called post-mortem degeneration. Plus some more information regarding the lysosome. Lysosome is called the suicidal bag because of autolytic role. It is also called disposal unitis, as it cleans the cell from the foreign agents. It cleans the cell from foreign agents. And so far as the membrane, integrity of the membrane is concerned, its integrity is maintained by the membrane of lysosome is maintained by cholesterol and cortisol. Once again I will recap. The lysosome It is a cell organelle found in case of animal cells, protozoans, in some plant cells, and it is also found in the fungi and absent in case of prokaryotes and mature mammalian RBCs. And then it was discovered by Christian Dido in 1955. There is a lysosome term that was given by Norkoff, and the number of lysosomes varies from cell to cell and from species to species. Their size is from 0.2 to 0.8 micrometer, and their shape is spherical in nature, and it contains about 40 enzymes. They have hydrolytic nature, and they are these enzymes I have mentioned on the board, and they are collectively called the acid hydrolysis because of the acidic medium. They work in acidic medium. Injured cell, if injured cell is there, the membrane of the lysosome gets ruptured, hydrolytic enzymes comes out and kills the nearby cells. That's called the post-mortem degeneration. And the lysosome is also called the suicidal bag because of the autolytic rule. They are called the disposal units because it cleans the cells from foreign agents, and its membrane's integrity is maintained by the cholesterol and the cortisol. I hope that you got this information. Another part of this topic is that is called polymorphism of lysosome. Lysosome shows polymorphism, means lysosome shows many morphological shapes. On the basis of function, the lysosomes are 4 types. The 4 types are in the form of, first one, primary lysosome, secondary lysosome, tertiary lysosome, and autophagy. So far as these terms are concerned, I'll mention the definitions of these terms in the description box. You can see the description box, and there I will mention their terms. Here I'll only explain on the board. So I will explain it with the help of drawing the diagram. This is the part of the cell. Dear students, in this diagram, I'll show the polymorphism of lysosomes. Means the lysosomes are of various types because of they play role differently. On the basis of that, here is a term called the primary lysosome, secondary lysosome, tertiary lysosome, and autophagy. 4 lysosomes play role differently. I have drawn here a structure of the cell. This is the plasma membrane of the cell. Plasma membrane of the cell. And I have mentioned here certain types of the cell organelles. The first one, this cell organelle, this is called the endoplasmic reticulum, and this portion of the endoplasmic reticulum, as you already know, it is called the rough endoplasmic reticulum. It is responsible for the synthesis of proteins, and this is called the smooth endoplasmic reticulum. This is responsible for the formation of lipids. Both lipid formation, protein formation, when takes place, then the proteins or the lipids goes inside the lumen of the endoplasmic reticulum. Then little bit of the packaging or the processing takes place. Then there is the formation of the vesicles that comes out. I have mentioned with the help of the arrows. This contains the material which is not fully, or we can say the 100% processed, or we can say the packaged. Then this vesicle comes in contact with the membrane of the Golgi apparatus. I had told you Golgi apparatus have 2 phases. One is called the convex phase. Through convex phase, there is the attachment of the vesicles which comes from the ER. I hope that you are getting the point. Then the processed material comes out through the concave side of the Golgi apparatus, and some of the vesicles gets detached from the endoplasmic reticulum and gets released from the cell as such, like this arrow, or the vesicles comes out from the Golgi apparatus and comes out from the cell by a process called the secretion. The second point, what is primary vesicle, or we can say the primary lysosome? Primary lysosome gets detached from the concave side of the Golgi apparatus. This is called the primary lysosome. Contains the powerful enzymes which have a role, which play a role in digestion of the macromolecules. Likewise, what is secondary lysosome? Secondary lysosome, this is called the secondary lysosome. Secondary lysosome. It is also called a heterolysosome. Heterolysosome. And this heterolysosome, it is also called heterophagosome. It is called heterophagosome. Why it is named differently, I will discuss here. The first one, the material goes inside the cell by a process called the endocytosis. If this material is solid, then the process is called phagocytosis. Entry of solid material inside the cell, that's called the phagocytosis. And when it goes inside the cell with a bit of plasma membrane, that is called a phagosome. Likewise, when there is the entry of liquid material from the outer medium into the inside of the cell, into the interior of the cell, the process is called pinocytosis. Pinocytosis means the entry of liquid material into the interior of the cell. Then there is the formation of a pinosome, little bit body inside the cell, that's called the pinosome. Pinosome contains the liquid material, phagosome contains the solid material. They approaches towards the primary lysosome because primary lysosome contains the hydrolytic enzymes or the powerful enzymes. These materials approach towards it because they have to get digested inside the lysosome. So this phagosome or the pinosome along with the primary lysosome form a secondary lysosome or the heterolysosome or the heterophagosome. I hope that you got this information. So primary lysosome contains only and only hydrolytic enzyme. Secondary lysosome contains the phagosome or pinosome along with the hydrolytic enzymes. Then what happens? Digestion of the material takes place inside the secondary lysosome. Then what happens? Undigested material remains after the digestion and it forms third structure, that is called the tertiary lysosome. I'll mention here, this is called tertiary lysosome. It is also called a residual body. So residual material contains the toxic substances or the material which are not needed for the cell to perform different kinds of the activities. Hence residual body or the tertiary lysosome must outside of the cell by a process called exocytosis. It is also called ephagy. Must remember what ephagy is. Ephagy means removal of the waste material in the form of a residual body from the cell. But there are certain cases when the residual body doesn't go outside of the cell because of many reasons. The first and the prime reason must be there is a lack of certain hydrolytic enzyme which are not found inside the cell, so they remains inside the cell. The residual material when remains inside the cell, it creates certain kinds of the problems collectively called as storage disease. Means when there is the accumulation of the residual body inside the cell, what happens? There is a disease called hepatitis. There is a disease called Hurler's disease. There is a disease called the polynephritis. And there is a disease called the Tay-Sachs disease. Even when there is an accumulation of the residual body inside the cell, it also leads aging. It also leads aging. I hope that you are getting the information which I am keeping in front of you. Once again recap. Primary lysosome only contains the hydrolytic enzymes. Secondary lysosome contains the phagosome or the pinosome along with the hydrolytic enzymes. Tertiary lysosome is the residual body that remains after the digestion of the food, means residual body, unwanted material that must be get rid of from the cell. The process called the exocytophagy. If there is the accumulation of the residual body inside the cell due to the certain reasons, one reason among them, that is absence of enzymes. The diseases are called the storage disease. Among them, there is a hepatitis, there is Hurler's disease, there is polynephritis, there is Tay-Sachs disease, and aging is also occurs due to the accumulation of the residual material inside the cell. Then the third and fourth and the last type of the lysosome, that's called the autophagy. And what is autophagy? Autophagy means there are certain cell organelles which are non-functional at the time of aging, means worn out cell organelles. Worn out cell organelles are wrapped by the number of lysosomes. Then the worn out cell organelle is killed, or we can say it is degenerated by the hydrolytic enzymes which are which wraps the cell organelle, which is worn out cell organelle, and it is responsible to kill it. That is the reason the lysosomes are called the suicidal bags, means they help in autodigestion. So this structure is called the autophagy. This red portion here I mentioned, it's a worn out cell organelle, worn out cell organelle, and these wrapping structures are called the lysosomes. These are the lysosomes. So autophagy leads self digestion, or we can say the digestion of material. So we say lysosomes are called the suicidal bags. I hope that you are getting the information. And now the functions of lysosomes. When we talk about the functions of lysosomes, it helps us to digest the unwanted material. I'll mention here digestion of unwanted material. First one, it helps in digestion of unwanted material. That means the material which is not needed for the cell. Unwanted material means the foreign agent comes in contact with the cell, that enters into the cell. Then the hydrolytic enzymes play a role in get rid of that material out of the cell, unwanted material. It helps in the digestion of useful material, like there is the entry of food material in the form of a phagocytosis and the pinocytosis. These are the useful material. These are digested by the hydrolytic enzymes. Another point, third point, it helps in autolysis, means breakdown of the material which is worn out cell organelles are found inside the cell. So it has autolytic role. Worn out cell organelles gets killed or digested by the hydrolytic enzymes. Another point, it helps in the fertilization. It helps in fertilization, means the lysosomal enzymes or the hydrolytic enzymes breaks the barrier of ova. Then what happens? Entry of male gamete takes place. Hence fertilization takes place, zygote formation takes place. Means there is a barrier outside of the egg, or we can say the ovum, that is broken down with the help of the enzymes which are found in the lysosomes. And another point, thyroglobulin gets converted into thyroxine. Thyroxine is a hormone which is found in the thyroid gland. It is found in the inactive form in the form of a thyroglobulin. Thyroglobulin changes it into the the hydrolytic enzymes changes thyroglobulin into the thyroxine hormone. Then it works. So far as the autolytic or autolysis, or we can say the autolytic role of the lysosome is concerned, it is responsible for the cutting of the tail in case of tadpole larva, because the tadpole larva gets changed into the frog so far as the metamorphosis process is concerned. So tail has no role because the frog has developed the limbs. So tail gets degenerated, or we can say destroyed with the help of hydrolytic enzyme. That is the that's the process called the autolysis. Another function of the autolytic role of the hydrolytic enzyme, so far as the we can say mammals are concerned, there are teeth, we can say the last molars, that there is a tearing of the gums takes place, and then there is a exposure of the last molars takes place. Okay. These are the functions so far as the lysosomes are concerned. I hope that you got the information so far as the lysosomes are concerned. If you feel that this video is helpful for you people, do subscribe my channel, hit the like button, comment in the comment section, and essentially share these videos with your friends so that they can also get the information so far as these videos are concerned, which I am uploading instantly on the YouTube. Thanks for watching the video.